Microscopes have changed enormously over the centuries. The light microscope was first invented back in the 1590s. The light microscope opened up a whole new world to scientists, where they could see things like the nucleus of an animal cell and chloroplasts in plant cells. This one here was used by a scientist called Robert Hooke back in 1665, where it uses lights from an oil lamp to help see the specimen. Hooke looked at some cork and drew a picture of what he discovered. Hooke noted the small holes within the cork looked like tiny room-like structures, eventually coining the term cells. You must know the definition for magnification and resolution relating to microscopy. Magnification is the number of times larger an image is compared to its actual size. For example, if a specimen is magnified by 100, then the image is 100 times bigger than the specimen's actual size. Resolution, on the other hand, is how blurred an image is. Therefore, resolution is the ability to distinguish between 2 points that are close together, such as 2 cells or 2 organelles. You must know the different parts of a microscope, so let's start at the top and work our way down. The eyepiece lens is what you look through to see the image, which is also magnified, usually by 10. The handle is, well, a handle. You can carry it. The objective lenses are what really magnifies the image. They are usually times 4, times 10, and times 40. The clips hold the slide in place. The stage is where you place the slide. The focusing knobs move the stage up and down, with the coarse knob moving the stage into place, then the fine knob to carefully bring the image into focus. Finally, the lamp, which shines through the slide to see the specimen more easily. You must also know how to prepare a specimen to view it under a microscope. Here's how. To allow light to shine through your specimen, you will need a small slice. Therefore, cut a thin slice of your specimen. Next, you will need to ensure your specimen stays in place on the slide. So pipette a small drop of water or mountant into the middle of the slide, securing it with tweezers. To see your specimen clearly, it is useful to stain it. Depending on what your specimen is, dictates the stain you use. For example, methylene blue is used to see animal nuclei, iodine is used to see plant nuclei, and crystal violet is used to see bacterial walls. Our example here is of plant tissue, so add a drop of iodine on top of the specimen. Place a cover slip over the specimen by gently lowering one side at a time using a mounted needle to ensure no air bubbles get trapped. Here's how to correctly use the microscope to view your specimen. Clip your slide onto the stage. Select the lowest powered objective lens. Not looking through the eyepiece, move the stage upwards until it nearly touches the objective lens. Then, using the fine adjustment knob, carefully move the stage downwards to bring the specimen into focus. Finally, to see the specimen with a greater magnification, repeat the same stages but with a more powerful objective lens. Your GCSE contains some maths, which are easy marks once you know what you are doing. The examiners may ask you to calculate total magnification. This is just the multiplication of the eyepiece lens and the objective lens you are using. For example, if your eyepiece lens is times 10 and the objective lens you are using is times 40, then the total magnification is 10 times 40, which equals 400. Easy. Just remember to be sure what eyepiece lens and objective lens they are asking you for in the question. To calculate the image size, actual size or magnification, you need to use the following equation. I remember it by calling it the I am equation. Firstly, identify what the question wants you to calculate. For example, calculate the actual size of the specimen when the magnification is times 100 and the image size is 7 mm. In this question, it tells us to calculate the actual size, so we know to divide image size by magnification. We put the known numbers into the equation as shown. Before we can do this calculation, we must convert the image size units into micrometers first by multiplying by 1000. This changes it to 7000 micrometers. Finally, we then divide 7000 by 100, which gives 70 micrometers. Do not forget the units at the end, as you may be deducted a mark.